the last section in chapter 16 is about how we package DNA. It's very short, very simple. Basically, you know that all your DNA is stored inside of your nucleus, and at times it's stored and looks kind of like a bowl of noodles, and at other times it's stored to look like chromosomes. And the reason for this is because as we really tightly coil our DNA into our X's, that way we can easily separate them during mitosis and meiosis, it's hard to access them to make proteins. Next chapter is all about making proteins, so we're not going to talk about that much now, but we can't get in there, read the DNA with our enzymes if it's really tightly packaged. So that's why we have two different forms of DNA. And it talks a little bit about bacteria. So basically, bacteria have no nucleus, but they store their DNA in something called a nucleoid, which is super tightly bound DNA. And this is how, so chromatin is what all this DNA and protein packaging is. So our DNA is never found alone. It's always held together by proteins as well. And chromatin is how we found it inside the eukaryotic cell. Nucleoid is the structure, since they don't have a nucleus inside of prokaryotic cells. Okay, this is the important stuff. So basically, here's our double helix. You guys should recognize that. It's about 2 nanometers in diameter. And then what happens is these things called histones, so you should know what histones are. They are proteins that help to organize DNA structure. They come along and they kind of wind around the DNA, and a bunch of them associate with each other, and we get this thing called a nucleosome. It's just like a little dot along the DNA double helix. And what it does is then it just allows all these nucleosomes to come together and to tightly be wound up. And it just helps all the DNA from getting tangled and allows it to be really tightly together. And so here's, it continues, and so here's all those nucleosomes together that are tightly wound together, and then they start looping together and they form a chromosome. So the important thing here to know is that DNA is never alone. It's always controlled, or it's always organized by these histones and the histone complexes, but it undergoes a lot of changes throughout the cell cycle. So as you know, during interphase, it looks like a bowl of noodles, like chromatin, but then during mitosis and meiosis, it condenses into our chromosomes, like we saw somewhere here. But when we're still in interphase, so it's still in chromatin, it's not always the same. So it's always changing. It's never static. It's never staying the same. Basically, if it's really loosely packed, this means that it's not all really tightly crinkled together, and that means that our proteins to go through transcription and translation and to turn that genetic code into a protein, our enzymes, our machinery for that, can get to the DNA. Think of it this way. If you really wanted this one specific piece of yarn, it's 1 inch, you needed that 1 inch, and it were like super tangled up in a ball of yarn, it'd be pretty hard to get to. But if it was in a nice loose braid or a nice loosely wound around something, you would easily be able to find it. And so that's what euchromatin is. Euchromatin is when we can easily access the genes that are found there because it's loosely packed. Then we have heterochromatin, and these are sections of DNA that we don't need to get to. There's no genes on there that we want. This is probably what our bar buddies look like, and they're just super tightly packed so that our DNA machinery can't get to it to make proteins out of it. And other places you would find heterochromatin are your telomeres on your chromosomes, because remember, telomeres do not code for anything. They are just there to prevent our DNAs from disintegrating or from deteriorating. And so in this case, heterochromatin is found in the telomeres because those telomeres don't code for anything. We don't need our machinery, our enzymes to be able to get to that because we're not making genes out of the telomeres. Same things with the centromeres. We're not making genes out of the centromeres, so they are usually heterochromatin as well. And that's what you need to know.